We're going to take a slight shift now. It's my absolute pleasure to introduce to you our next speaker, Dr. Richard Turner. Richard worked for a number of years in Liverpool with Minir as a clinical pharmacologist, and more recently has gone to work in industry for GSK, and is very kindly agreed just to give us a perspective on some of the needs and the uses of pharmacogenetics in that context. So, Richard, thank you.
Hi, everybody. Thank you for the welcome invitation. It's great to be here today. So, yes, as mentioned, with a little bit of maybe poetic license, I'm going to try and share a scientific perspective as we see it on the uses and opportunities of genetic and genomic data linked to healthcare data, real-world healthcare data, to inform drug discovery and development, to ultimately benefit patients and improve clinical outcomes. I will also deliberately, and I say this with a little bit of hesitation, but I will deliberately try and save time at the end for questions. I might try and speak for maybe 2/3 of the time. And I do that because as a clinically active researcher within a big pharma company, I am conscious that I only represent a tiny slice of the breadth and depth of industry that may be interested in pharmacogenetics and potentially interacting and engaging with the network. And therefore, it would be great to have questions, discussion at the end, and to hear the viewpoints of others. So, one of the, obviously, the major things we think about in industry is the attrition rate. How can we improve our probability of technical and regulatory success? And this is so important, it impacts all of us, because as the number of programs that do not make it through to regulatory approval increase, the costs for each drug that does make it through that net also increase, and that has impacts on all of us and our healthcare systems. At the moment, still, for assets that make it into clinical development, only about 10% of those at phase 1 will eventually succeed, so you have a 90% attrition rate. And the reasons for this have evolved over time, but at the moment, one of the major, the largest reason is demonstrating efficacy. So that's both proof of concept that it works, and then that it is superior to current standard of care, which obviously is also, the bar is rising over time, which is a good thing. So why is this, and how can genetics possibly help? Fundamentally, it's because correlation doesn't always equal causation. You can find lots of observational associations with disease susceptibility, with different severities, from all sorts of different biological molecules, and therefore you can formulate many different therapeutic hypotheses. But if it's what you're targeting is not causal in that disease pathogenesis, then it's unlikely to improve clinical outcomes. So it has been noted, and many companies now building on this of more recent research are utilizing some of the unique attributes of genetics to be able to unpick this causal inference question. The fact that our genetic variants are inherited at birth, we receive them at conception, and they are randomly assorted to us. That means that we are free from some of the common biases, such as reverse causation and confounding, selection biases, and therefore being able to detect signals that a genetic variant is associated with a particular phenotype can give us some hypotheses that we might be able to identify targets which might have a causal bearing on that disease. And the plot on the left just demonstrates that not all genetic variants are equal. This is also an evolving space. Where you have PLOF, so sort of predicted loss of function variants, which are generally rare, but they're exonic and sit in the gene, those variants, when they are associated with disease, they perhaps give us the greatest confidence from a genetic perspective that we may be onto something here. it's moving though the whole time. So missense variants, EQTL, sometimes being able to figure out which of the genes, particularly those EQTL variants, map to is part of the challenge. But as a suite of new tools over the last decade to help to influence drug discovery and development, I'd say genetics has probably been one of the landscape changers in big pharma. So what are we going to talk about? So I think genetics and how it links to healthcare data can impact right across development from preclinical to clinical. And actually this does not include sort of subsequent life cycle management, indication expansion, pharmacovigilance of assets that have recently been licensed. But I'm going to touch on three things. A little bit more on target identification, then maybe how we could use genetics in some ways to help understand mechanism and disease understanding, and then talk about pharmacogenetics. And once we've developed drugs and we're testing them and what we can do there. So this is it's a colourful PheWAS plot just to illustrate that this is, I think, for a loss of function variant for IL-33. So this is just to show that across the disease or phenome spectrum, you can find, and I didn't know we were talking with asthma just before this, but you can identify variants and therefore genes, and then look at both their associations. You can do that with binary phenotypes, you can do it with intermediate phenotypes, and this is one. So IL-33 variant, peel-off, it associates with eosinophil counts, and then it associates with a variety of different atopic diseases, and asthma's at the top of the list there. So it won't surprise anyone to know that there's an awful lot of clinical development happening in this space across a variety of different indications, and eosinophils look to be fairly important. So that's just an example. One of the other uses for this is being able to think about safety from an early point of view. And actually then sample size is critical, because the absence of evidence is not always the same as evidence of absence. But if you're up in the hundreds of thousands, millions of cases, and you're not seeing a particular association with a phenotype that might be of concern, it doesn't necessarily prove that the drug, which might modulate your target to a greater extent than your genetic variants, will be free of particular safety concerns, but it can help to inform that decision making. Most of the genetic work is done comparing cases and controls, but generally we treat patients and we're trying to prevent progression, we're trying to prevent severity of that disease. Occasionally genetics can also help in that case-only setting. So this is an example on the left of missense variant in a gene called MARC1, which is associated with a spectrum of different non-alcoholic liver diseases. It should now be MAFLD. That's comparing susceptibility cases versus controls. But actually, if you go within those people who already have liver disease, you can have a sort of survival plots for this particular variant and demonstrate that those who are homozygotes for the missense variant, they have a reduced risk of liver-related mortality. And you can imagine when you're trying to engage stakeholders, other people from across an organization that may not have so much familiarity with genetics and pharmacology, that being able to say that it associates with susceptibility, but also actually we are seeing associations with hard clinical outcomes in a case-only setting, that can be quite impactful. All of this, though, requires genetic and genomic data linked to healthcare records. And I would say probably the UK is, can we bias it, probably a world leader in this ability to generate population-based and disease-based cohorts and enable access. And I was just at a meeting yesterday where Our Future Health is celebrating that they have what they're terming a fully recruited over 1 million participants now. So it's great to see the interest, the collaboration, and these cohorts really driving this vibrant part of the UK life sciences sector forwards. And so this is, this is perhaps not the most aesthetically pleasing slide, but when I was writing this a bit in one night, I was thinking of some of the attributes, and you've got both population-based, you've got patient-based, and you've got, for example, Genes and Health cohort, which is particularly focused on individuals who are UK, but of Bangladeshi or South Asian origin. which is very important, again, to sort of increasing the diversity in our data sets to help understand ultimately what's the full spectrum of genetic variation that we can see, and to help to ensure that what we're producing is going to be applicable to everybody. We're not just going to target one socio-demographic. So that's around sort of target ID, but some of those cohorts I mentioned there, NIHR Bioresource, and there's others as well, they enable recontact, and you can recontact individuals based on phenotype, so healthcare measures, as well as genotype. And this is novel, this is interesting. You can envisage that you might want to recontact individuals based on a polygenic risk score, just at the extremes of those, because that may be a more efficient study than just doing a blind recontact approach, where you're going to be better powered in order to try and detect differences for a given sample size. Or you can just focus in on a given one or a small number of genes to again try and recruit those two contrasting homozygote groups. So what's been happening in this space? I mean, I put two little examples here. So one was around apolipoprotein C3. This is a few years old now, but it's a heterozygotes for a peel-off variant in this were associated with decreased risk of cardiovascular disease, which is interesting, but there was a slight concern. Well, why haven't we seen this with any homozygotes? Could that be a possible safety risk if you entirely lose the function of this gene? And this was done in Pakistan. They found a sort of probanded index case that did appear to be a homozygote, and actually could recontact them, and then went to different family members. And if I recall correctly, the spouse actually by chance also happened to be a homozygote. So there were several members in this family who were either heterozygote in the extended family or homozygote within the direct family, and they were able to demonstrate that these homozygotes compared to normal or heterozygotes had significantly decreased levels of apoprotein C3 and a sort of postprandial blunting of triglyceride concentrations, which again might therefore be indicative of cardiovascular protection. And then on the bottom, this came out early this year. I think I saw it on the BBC first, actually. So there was I did go to read the paper afterwards, but this was a great paper. Lots of work had been done looking at intergenic gene harbor. I think it was a gene desert region, I think chromosome 19, which had been strongly associated with a variety of different autoimmune diseases, inflammatory bowel disease, I think ankylosing spondylitis, Takayasu's, primary biliary cholangitis, if I recall. And the question was, well, what's the mechanism? Which is the gene? And so they conducted a panoply, I should say, of functional genomic investigations, and it included actually recontacting some individuals once they'd homed in on the gene from the bioresource who were homozygote for this particular variant, and then they were able to demonstrate difference in the gene expression of genes of interest within resting macrophages, and all this helped to build up a picture of what this region was doing and just how important and instrumental it is to several of these diseases. It appears to be quite upstream, so we'll have to watch that space, what happens next. Lastly, pharmacogenetics. So obviously, clearly, that this is another huge area for use for genetic information and opportunities. And as we all know, this is the study and application of the genetics to optimize drug response. It is one of several factors which often interact with one another, which affect drug response outcomes. And this is important. We all know this because inter-individual variation, unfortunately, leads to differences in response. It leads to hospitalizations, prolongation of hospitalization, probably non-adherence. I'm glad to say, I can see as I sort of that these established drug gene pairs are iteratively, incrementally, being put forward for PGX testing. And clopidogrel 2Z90, I'm sure will be discussed, being nice and also it's great to see this moving. Mentioned, as we know, there's many really strong associations with the HLA region, in particular idiosyncratic adverse drug reactions, and whilst maybe the positive predictive values limited, these got a high negative predictive value, so there's still utility here, perhaps around the diagnostic space. But it's not just established common drugs that for which we have interest in pharmacogenetics. This is, again, maybe a slightly detailed slide, but just of some of the more recently licensed drugs where during licensing pharmacogenetics has been part of the regulatory approval process, from Illusius, where you do need to do 2D6 pre-treatment genotyping. Similarly for CYP2C9 with saponimod, and that's for secondary progressive MS, and most recently for mavacamten for hypertrophic cardiomyopathy for pre-treatment 2C19 genotyping. And I mentioned one from the US here for brexpiprazole, and just to highlight there that on that label, as far as I recall, it doesn't mandate it. This is part of the challenge with labels, but it says if you're known to be a poor metabolizer of 2D6. And furthermore, the complexity is increasing. These labels are not just talking about a drug-gene interaction. They're actually also sort of drug-drug gene interactions, because they're also giving advice of what to do if somebody's taking a concomitant inhibitor, say, of CYP3A4 as well as having a particular genotype of relevance here. So good to see, but again, in order to scale this up and to implement it, it's going to need healthcare clinical decision support to help advise and assist us here. What else is emerging? So this is, this is in the Alzheimer's space for aducanumab. This did receive accelerated approval, I think, 2021 from the FDA, along with, I think it was a lecanemab, there was clearly lots of discussion around this, but these monoclonal antibodies which target beta amyloid in order to try to reduce that beta amyloid load in patients with Alzheimer's disease have been at the vanguard of efforts of late to try to improve the horrendous disease burden of Alzheimer's. Unfortunately, some of these drugs have adverse drug reactions, and one of the most recognized is ARIA, amyloid-related imaging abnormalities, and it's been recognized that this is associated with ApoE gene status, which is also the gene of greatest risk for spontaneous Alzheimer's disease. And this was a case-only GWAS of a couple of the phase 3 trials that previously done for aducanumab, and they confirmed that ApoE appears to be, and particularly E4 homozygotes, appear to be a significantly increased risk of ARIA, and there's different types, so edematous ARIA and what you call ARIA-H, which is either microhemorrhages or superficial siderosis. This paper did also show a nominal association with an Alzheimer's disease polygenic risk score, which perhaps suggests that there are other genetic variants. It's not just going to be ApoE which are associated with ARIA, but it perhaps wasn't powered enough to detect them with suitable statistically significant robustness. So obviously, that we have to watch where the drugs go, but I think this is indicative of, GWAS and polygenic risk scores, as well as thinking about candidate genes, all playing a part in analyses of our clinical trials, or clinical trials from a variety of different companies. And, I know we've had mention of polygenic risk scores, but this is a few years old now, but for some of the PCSK9 inhibitors, when they were tested in phase 3 trials in the secondary prevention setting for cardiovascular disease, again, it was demonstrated that a cardiovascular polygenic risk score appears to associate not just with incident disease, but with trial endpoints, in this case, major adverse cardiovascular events. And those patients in the trial that had the greatest genetic polygenic risk scores, they appeared to derive the most benefit compared to other participants with lower scores, and particularly those sort of in the 90th percentile and above. But it appears to be prognostic on the whole, at least when I look at this. In other words, it's the placebo arm. If you've received placebo and you've got that greater genetic liability for these diseases, you do worse, and you're getting a course correction with receipt of PCSK9 inhibition. But perhaps there are, some interest in, as well as sort of prognostic utility, but actually using genetics, I should say sort of multi-SNP polygenic risk scores for predictive utilities. So this is some work, it's retrospective, we'll have to see where it goes. I think it was the 44 SNPs have been identified, made into a sort of polygenic response predictor score, which interacts with beta blocker exposure in patients with heart failure in order to influence mortality. And the suggestion is that it's only those who both receive beta blockade, a suitable dose, and have a particular score below a set threshold that have improved survival, whereas the other three groups, either those who don't receive beta blockade or do but have the highest higher level of this genetic score, they appear to all track and have the same sort of degree of worse survival. So we'll have to see where this predictive rather than prognostic score goes, but it's really promising. And so just to finish off my last slide, you know, I was sort of thinking around what are some considerations from where I sit at the moment. And I sort of divided it along the lines I've been talking into three areas. So, you know, biobanks and cohorts that we're using perhaps in that target discovery space. Sample size is king, really important, but it's not the be all and end all. It's the depth and the breadth of that health-related phenotypic information, the primary care, the secondary care is key, and it's samples as well, and the ability to produce intermediate phenotypes, transcriptomics, proteomics, etc. That's going to become increasingly important. Recall by genotype studies, I've provided a few examples where it's worked. I mean, these are challenging. If you've done it, these are challenging. You have to think first and foremost about the participants, their well-being. We don't want to lead to recontact, fatigue. If you're just going to a single site, maybe that's doable. But what if these cohorts are nationwide and your participants of interest are sitting in multiple different NHS trusts or sitting in multiple different geographic locales? How do you operationalize that? I'd love to hear and talk about that more. And then thinking around the PGX side and real world data, you know, it has struck me that primary care data, we have our FBC, liver function test, using these. One day we'll maybe have, you know, routinely, as it gets rolled out, you know, pharmacogenetic testing. And that will be great for those patients, but it will also then feed back into these research uses, these cohort uses. So we'll be able to increase our real-world evidence for those drugs to look at novel associations with other commonly prescribed drugs. Is it going to help for drugs in development? I suspect so, and for pharmacovigilance, definitely. But again, there are challenges when we think about the drugs. What about drug-specific phenotypes? You know, ICD-10 codes, SNOMED codes. There are things that are missing. And even the prescribing data, when we think, you know, GPs obviously do the bulk of the repeat prescriptions, but prescriptions which are initiated in hospitals, we don't always have access to those, for example. So there's things that are missing. Clearly, though, there are also novel approaches here to think about PGX, such as the MRHR yellow card biobank, and its focus, I believe, on DOACs and allopurinol side effects, as well as, again, as a novel approach in order to start to link up recontacting essentially are based on adverse drug reaction, and then to generate that sequence data and to make that available to researchers to link healthcare to genetic to genomic data in our vibrant ecosystem. So those are my thoughts there from a slightly somewhat different perspective. Happy to take any discussion questions now, and I'm here for the rest of the morning.
Thank you very much, Richard. Really helpful and very nice overview and perspective. Any questions from the audience? I'll give you a little time because I'm going to start off, because obviously you work for GS. Yeah, take a seat, make yourself comfy. Yeah, you obviously work for GSK, which is a huge organisation, and there are sort of, you know, a whole spectrum of pharma companies who have got different resources available to them. I know there was a time when a number of the big pharmaceutical companies particularly invested really heavily in pharmacogenetics, had their own units where they were undertaking that work, whereas smaller companies would maybe outsource that to maybe some of the providers and people who would be represented in this room. can you give us a sort of sense of what the sort of landscape is at the moment and the appetite for undertaking these sorts of studies?
Yeah, sure. No, great question. I'd say the landscape is still probably, as you'd imagine, heterogeneous and mixed. Some companies are very invested, others less so. Again, that's partly pragmatic and based on resources. I think there is some push and pull, though. If you are developing a drug and you find out in vitro quite early on that its metabolism or transport might be dependent on an established pharmacogene, you can envisage that PGX will probably have to be part of that development at some stage, and I think that probably goes for all developers. There are unique challenges to rolling out genetic testing in international multi-center trials. We have a wonderful setup in the UK here, but attitudes towards genetic testing, data privacy and so forth varies across the world, and therefore even to be able to perform genetic analysis within a given phase 3 trial takes quite a lot of work under the hood, actually.
Yeah, that's really interesting. It's something that we sometimes have a bit of a UK perspective, but you're working in an international sort of environment and understanding those complexities. One of the things that sort of links with that is that sense of, I know very much in the cancer space, the development of some of the sort of targeted personalized medicines, there's been a sort of co-development around companion diagnostics, working with pharmaceutical companies. Is that something that you get a sense that there's an opportunity or an appetite from within pharma around pharmacogenetic tests? As you mentioned, some of the new examples, mavacamten, siponimod, these sorts of things. There's a sense that actually the drug is pushed out there, it's the regulator saying, oh, you should do pharmacogenetic testing, and then people do that, or it's the companies thinking as they develop it that we need to think of a way as we launch it that we co-develop that.
No, good question. I think I didn't go into oncology on those slides, but you're right. Again, oncology is the poster child for many of this precision medicine work. So it's recognized and established the use of companion diagnostic CDX within the clinical space, and therefore I think that makes so much difference to early development, because they know that if you have that companion diagnostic developed in parallel, that will be able to be implemented in clinical practice, and that's really important. You cannot overemphasize that enough. For other things such as germline PGX, where that is not yet the case, you can imagine that there is worry and concern about how is this going to be received, how is it going to be implemented, who's going to pay for it, and so on and so forth. So again, I think as more and more things get rolled out for the existing drug gene pairs, it will smooth the road. It will show how implementation can happen. Ultimately, though, it is anchored on clinical utility, and part of development, it's a lot of work to develop a companion diagnostic. So part of the challenge is if you have to have a really good idea of it when you're going into your phase 3 trial. So with oncology, if you know it's the target, you will maybe already have your assay, your CDX set up by phase 2. It's been proof of concept tested in phase 2. You get it good to go into phase 3. A lot of this PGX, for example, I showed you for aducanumab, that is retrospective exploratory testing at the end of the phase 3s, and that's too late.
Yeah.
For it. So then you need to go embark on a whole new course. So that's part of the challenge, and I think we all need to work together, industry, healthcare, academia, regulators, to sort of figure out how to solve that problem.
Yeah, really interesting. Thank you, Richard. Munir.
Richard, so this is a difficult question.
I defer. No.
At the moment, there's a model for drug development is very much based on populations, and therefore that's translated into summary product characteristics, which is also very much population-based. Right. So if you look at some of the statements that come out in terms of when drugs are being developed, it says the change in AUC is only 50%. So it's not relevant
Mhm.
at population level. But that doesn't mean it's not relevant at the individual level when you're giving it to hundreds of thousands of people.
Yeah.
We forget about the fact that it's actually affecting those individuals, which may be at the top end
Mhm.
of 50% or maybe even higher than that, because 50% of
Is
the population.
is it, yeah.
So how do we actually change that model of development to actually take into account individuals? Because as you know, in clinical practice, we're not treating populations, we're treating the patient in front of us in our surgery.
Yeah. No, again, a great question, Munir. No, you're right. You see a confidence interval around that 30-40% increase in AUC, and at the population level, it may not be any more impactful than any other clinical variable, but there are individuals at the extremes of that where, for other reasons, that given increase in AUC with their other factors might be clinically relevant. I think we just have to work in partnership to do that. I sort of view, even during clinical development, you've only exposed a limited number of patients to the drug, and therefore a lot of, it goes into the real world, a lot of activity has to happen afterwards. We are, and I guess, safety is really essential. So if any sort of safety signals are flagged up, that maybe it's coming almost from a phenotype approach, then these will get an in-depth review. But in order to sort of change that paradigm, it's difficult. They're looking for population-based efficacy and safety. That is the metric at the moment. To make that personalised, maybe the way to go is through trying to reduce it through precision medicine. To actually, if you build in the CDX to reduce those increases in AUC, or maybe it's through modelling. Population-based PK modelling, and can you then have simulated patients to try and demonstrate that. So it's really tough. You've got different interests and small sample sizes. I think I could still see a role. I really do still see a role actually for academia in newly licensed drugs in order to start to tease this out and unpick it. It's probably a bit of an unsatisfactory answer. It's very difficult. You said it was a difficult question.
Thank you, Richard. Final question just from Ron at the back.
Yeah, thank you so much. Perhaps I have an easier question then. I was wondering, you showed very nicely with the siponimod and mavacamten that there is now indications for 2D6 or 2C19 genotyping. I could not help but noticing that in all the 4 drugs it was stated that you should avoid 3A4 inhibitors. Yet there's already 14 years of polymorphism known in cytochrome 3A4. So I was wondering, does, for instance, GSK take that into account? Because, do not use inhibitors is very quickly stated without any randomized controlled trial, I think. So why not include the polymorphism in cytochrome 3A4? Could you reflect on that?
I think you probably speak to a broader thing there about, are there different standards applied? You could already take that for the implementation of genetics in the healthcare sector. Is the bar higher than what we use with routine liver and kidney testing? I think drug drug-interactions are very well recognised. The evidence underpinning them actually is perhaps less substantial than you might imagine, but they are recognised, they are established, and it feeds into the previous question. They are known to potentially dramatically increase exposure. Patients who are taking those other drugs might have other comorbidities, for example, and that can all feed into concerns, and therefore it is something which is established, easy to do in clinical practice. You avoid the drug, you halve the dose, so it's quite easy to action. For genetic variants, again, it's this whole catch-22 of even if we find this, how did it get implemented? How does it get actioned? It goes back to that. But obviously the variants, and I'd rather not work at a variant level, it'd rather be a sort of gene-based level, but the variants, again, we're still at the stage where I think you would need to demonstrate that the specific variants have, a significant effect on exposure, whichever those variants are. I think that would still be useful at this point in time.
Thank you.
Thank you very much, Richard, and thank you very much to all our presenters this morning for a really great overview of where we're at and some interesting areas. So thank you so much. It's now workshop time. If you, that's just changed. Yep, perfect. If you are interested in contributing, participating in the mental health workshop, then stay here. The other two workshops, am I right, are both upstairs?
The cancer workshop is upstairs, and the paediatric is just next door.
Okay, so if everyone heard that, paediatric with Dan is next door, and the cancer workshop with Sophie and Jess is upstairs. Perfect. And we're all coming back here at 1:45.
We